I'd like to welcome everybody. Good morning to our NCI CCR Liver Cancer Program seminar series. Today we have a seminar by Dr. Stephanie Ma. Dr. Shin Wong is going to formally introduce Stephanie, but I just wanted to note to everyone that we have two more upcoming seminars in our spring series. On May 18th, we will host Dr. David Brenner from UC San Diego, who is now president and CEO of the Stanford Burnham Presbyterian Institute. And on June 12th, we'll have Dr. Anna Mae Deal from Duke University. So I hope that you can join us for those seminars as well. We do have a fall lineup for these seminars as well. So we hope that you'll take a look at our LCP website for those seminars that we'll host in the fall and winter of 2023. So thank you all for joining us today. If you have questions for today's speaker, please put them in the chat and we'll address them at the end of the presentation. So Shin?
Wonderful. Thank you, Anu. And good morning, everyone. So I'd like to introduce our today's speakers, Dr. Stephanie Ma. Now, Stephanie obtained her Bachelor of Science in Cell Biology and Genetics, and then master degrees, experimental medicine from the University of British Columbia in Vancouver, Canada. She then received her PhD from University of Hong Kong. Now, since then, Stephanie has been working at the same university, where she's now the professor and associate directors of the Knowledge Exchange and Global School of Biomedical Sciences. Now, I actually came to know Stephanie from her work on liver cancer stem cells. She actually published among the first paper that convincingly demonstrate that the CD133 positive tumor cells as a cancer stem cell. Now, this research, I think, has set her apart from many others at the forefront on this research field. Now, since then, Stephanie has been continuously explore Stemness as a cancer cells vulnerability. Her work has been well recognized and has been listed at one point at the top 1% of most cited scholars under the category of clinical medicine and all field. This is very impressive. Now, as such, you can imagine that Stephanie has received numerous awards that includes the Young Scientist Award in Life Science from the Hong Kong Institute of Science and Outstanding Young Research Award from Hong Kong University. And then University of British Columbia Alumni Builder Awards, and among many others. So without further ado, I'd like to formally welcome Stephanie. Stephanie, the podium is yours.
Thank you very much, Dr. Wang, for the very kind introduction. And also Dr. Budu for the invitation to speak at this webinar series. Can everyone see my slides clearly? Yes.
Yes.
Great. Awesome. Okay. Thank you. So, so it's really my pleasure to have the opportunity to share with everyone some of my team's recent work on understanding phenotypic plasticity in liver cancer. And it's a really good opportunity also to meet individually with so many of this NCI liver cancer program members. So, as we all know, hepatocellular carcinoma HCC, it's a deadly disease with limited treatment options and a very poor prognosis. Now, contemporary challenge in treating cancer has really been the common therapy resistance and recurrence after therapy, all of which have been reported to be associated with stem-like behavior of cancer cells. Now, the hallmarks of cancer that was published by Hanahan and Weinberg, of course, in 2000 and 2011, has provided the original and updated conceptual framework of cancer cell biology. Earlier this year, Hanahan has also published an updated version that highlights additional emerging hallmarks and enabling characteristics, including those that are under the umbrella of unlocking phenotypic plasticity. Now, phenotypic plasticity is basically an acquired hallmark that that enables various disruptions of cellular differentiation, including dedifferentiation from mature to progenitor states, block differentiation from progenitor cell states, as well as transdifferentiation into different cell lineages. Now, there is good data to show that HCC tumors contain less differentiated and a more stemness state that are more resistant to therapy and associated with the development of tumor relapse. So my research group has, like Dr. Wang said, has been interested in the study of how phenotypic plasticity can contribute to therapy resistance and tumor recurrence. So this is a figure that is taken from a review article that we published last year that documents milestones of cancer stemness related discoveries in HCC with some of my own team's work cited. So in the earlier years, we contributed to the identification of a number of functional liver cancer stem cell markers, including CD133, aldehyde dehydrogenase, and along with our collaborator, Professor Irene, also of Hong Kong U, CD24 and CD47. Now, back 15 years ago, most of these markers have been identified by sorting of the cell surface marker by flow cytometry and then xenotransplanting them at limited dilutions into immunodeficient mice. But these methods have been debated because these tumors have to be dissociated completely and sorted into single cells for transplantation into severely immunodeficient mice, which do not really recapitulate the setting in humans. So more recently, in the past 5 to 8 years or so, there's been an increase in studies on the use of lineage tracing and lineage ablation coupled with single-cell RNA sequencing for the study of cancer stem cell origin and heterogeneity. And our team also recently published a paper where we utilized lineage tracing to investigate genetically marked CD133 positive cells to try to understand the role, the heterogeneity, and the properties of CD133 or prominent one, which is basically the mouse homologue of CD133 in a tractable HCC mouse model. So to start, we looked at CD133 expression in 2 HCC mouse models, namely the one that is chemically induced by DE-NCCL4, as well as one that is induced by injection of proto-oncogenes NRAS-AKT through hydrodynamic tail vein injection into C57 black 6 mice. Now you can see that PROM1 is progressively increased from early to advanced HCC, concomitant with alpha fetal protein in both models. And this was also validated by immunostaining, where you can see CD133 to be present in the tumor and not so in the non-tumor. Likewise, in TCGA human data, we were also able to see a stepwise increase of CD133 expression from early stage to late stage human HCC tumors. Now, then following that, 2 members of my team, Lina, who was a former postdoc, now a faculty member at Guangzhou University in China, and Eric, who is now a research assistant professor in my team, have started on a project where they wanted to use genetic lineage and genetic ablation to try to understand PROM1 in an intact mouse model. So they performed lineage tracing following visible tumor nodules formed in both the DE-NCCL4 as well as the NRAS-AKT model. And you can see that specifically 1 single dose of tamoxifen would be applied to the PROM1 TD tomato mice after the tumor initiated, and then we followed it for 30 days. So after tamoxifen treatment, the frequency of the TD tomato positive cells increased progressively from day 3 to day 10 to day 30, and the TD tomato cells were found in 50% of the tumors, indicating that the PROM1 derived TD tomato cells exhibited competitive amplification potential. We also did a low dose tamoxifen tracing, which labels only single clonal PROM1 positive cells. And you can see that there will be a clonal size expansion over the tracing period. And notably, the TD tomato positive cells did not overlap with keratin 19, but primarily overlapped with alpha fetoprotein staining, suggesting that they mainly represented HCC cells, but not the ductular cells. So on further characterization, we also treated the DE-NCCL4 mice with BRDU for short-term labeling. And where we could see that there was a higher percentage of PRDU that could be incorporated in the TD tomato positive cells, indicating that they had actually had a faster turnover rate. And the TD tomato positive cells also displayed enhanced Ki67, showing that they had enhanced proliferative capacity. We also went on to try to validate this at a functional level, and we showed that the TD tomato positive cells were able to reconstitute tumor organoids from single cells, and that they showed higher plating efficiency. We also transplanted sorted TD tomato positive and negative cells, or unsorted cells, into mouse livers through splenic injection. And you can see that as few as 1,000 TD tomato positive cells could give rise to HCC tumors. And this was almost 18-fold higher repopulating frequency than that of the TD tomato negative cells. So suggesting that these prominent positive cells really had a high proliferative and also functionally exhibited cancer stem cell behavior. We then went on to investigate whether prominent lineage could have any anti-tumor effect. So we then did a depletion model. I don't know if you can see it. How do I get? I don't know if you can see. Anyway, okay. So so we went on to do a depletion model where we crossed the prominent lineage mice with DTA. So you can monitor them by MRI, and you can see that for the control, there was be a 1 to 3-fold increase in tumor size over the baseline, as compared to only approximately 1-fold for the prominent depleted mice. Prominent depleted mice also had significantly fewer tumor nodules and smaller tumor sizes than the control mice. They also had reduced Ki67 staining, suggesting that they had a reduced proliferative potential. We also performed a similar depletion model in the NRAS-AKT model. And so we found the prominent depleted mice treated with tamoxifen showed a reduced tumor growth and prolonged survival when compared to mice of other groups. So we then went on to try to understand the lineage characteristics and molecular signatures of the prominent derived HCC cells. And so we did a single cell RNA sequencing analysis. Now, we found that, there were a number of clusters, of which clusters 1, 2, and 8 actually exhibited a higher copy number variation than the other population. So we identified these as HCC. So ultimately, we were able to annotate 8 major cell lineages in the population, including HCC cells, cholangiocytes, macrophages, monocytes, endothelial cells. There would be the hepatic stellate cells, fibroblasts, B cells, neutrophils, and also a cluster of cells called hybrid hepatocytes that express both hepatocyte and cholangiocyte markers. So we integrated the HCC cells from all 6 samples for analysis and found that the HCC subset to be mainly comprised of 4 subclusters that indicated a high degree of intratumoral heterogeneity of the HCC cells. So by RNA velocity, we were able to see a directionality from C3, C0, C2 to C1 in the TD tomato positive HCC cells, and this directionality was not evident in the TD tomato negative cells. We were able to also see that a number of hepatic differentiation transcription factors like HNF4 alpha, CPPA, as well as FOXA3, as well as their regulons, were decreased in C2 and C1 as compared with the C3 and C0, which suggests that they may represent the differentiation statuses. And in accordance with the pseudotime analysis, we would see an enriched positive EMT, regulation, proliferation, and embryonic-like signatures that were found in the C1 cluster as compared to the C3. So this data not only suggests that there would be a loss of differentiation phenotype, but also suggested a transition from a more proliferative to a transition to a proliferative and mesenchymal-like status along the trajectory. Most interestingly, if you can focus on subpanel F, we were able to see that transition of the PROM1 lineage HCC cells to share a similar transcriptomic profile with a human HCC differentiation grade. So you can see that C1 which is here had actually the highest correlation with the poorly differentiated HCC G4, whereas conversely, the C3, C0 subclusters actually had the highest correlation with the normal to low-grade HCC, and then C2 would be something in between. So here the data suggests that the highly heterogeneous PROM1-derived HCC cells follow trajectory of the differentiation during tumor progression, and these characteristics potentially contribute to the proliferation and also tumorigenicity of the PROM1 HCC progenies. We went on to try to decipher or to understand the mechanism facilitating the propagation of these PROM1 positive progenies. So we tried to identify a conserved marker that would be upregulated in the TD tomato positive cells across the 4 subclusters. So this led to the identification of 836 genes, which we call the PROM1 lineage signature. And if we use this 836 gene and apply it to human data sets, TCG, as well as the ICGC cohorts, and did unsupervised clustering, we can see that the signature high group would show a significantly worse prognosis than those in the signature low group, revealing that it means something clinically regarding the signature of the PROM1 lineage. And then we also did geo annotations where we found that the TD tomato positive cells were associated with strong anti-ROS and oxidation detoxification properties. So in the latter part of that study, but I didn't show the slides here, we did some functional validation where we found the TD tomato positive cells to also show significantly increased viability after ROS challenging. And so these collectively, the data shows that PROM1 lineage were more resistant to oxidative stress, and that this property may be a protective mechanism for rapidly propagating tumor cells that survive in HCC tumors. So in addition to looking at liver cancer stem cell markers and understanding them, we also spent quite a bit of work on trying to understand what are the intrinsic regulators or the extrinsic regulators of cancer stemness. So earlier on, we also tried to identify a number of deregulated genes, microRNAs, secretory proteins, etc., that will regulate stemness. We also, in more recent years, moved on to the cancer stem cell niche, where we identified how a soft extracellular matrix, how a glucose-deprived environment, or how cancer-associated fibroblasts can contribute to the maintenance of a more stemness phenotype in HCC. So today, I chose and selected a few studies to share, some of which is unpublished. So one of this study was actually published at the end of 2021 and done in collaboration with a colleague of mine in the School of Biomedical Sciences at Hong Kong U, who has expertise with CRISPR and in particular combinatorial genetics. So now, usually we like to do systematic testing of existing drugs. And we find that maybe combinations is an attractive strategy to try to repurpose some of the drugs for better treatment options. But instead of trying the drugs one by one, we wanted to expedite the search process and to also try to understand possible combinations. So we turned to combinatorial CRISPR-Cas9 screen to inhibit druggable targets related to stemness. So Alan came in to build a bar-coded CRISPR library of 30 plus stemness related genes that my team and others have found to be critical in maintenance of HCC stemness. So via this CRISPR screen, we were able to identify co-blockade of NMDAR1 along with targets FLT3 and FGFR3, which are targets of the first-line kinase inhibitor sorafenib, to be able to reduce HCC cell growth. Now, clinically, HCC patients with high NMDAR expression showed worse survival. And if we combine NMDAR antagonists, which is ivemprodil, we were able to synergize it with sorafenib to induce unfolded protein response, to trigger cell cycle arrest, and also to downregulate gene expression associated with Wnt signaling and stemness, and reduce self-renewability of HCC cells. So basically, we were able to show this in cell line models, in patient-derived organoids, as well as in mouse models. And because ivemprodil is actually approved in the clinic for use in humans as a vasodilator, we know that it should be safe, right? So our findings here support the repurposing of this drug as an adjunct for HCC treatment to improve clinical outcome for sorafenib resistant patients and also reduce tumor recurrence. So a separate study that is actually still unpublished, and we hope to be submitting this work soon, it's on chemo resistance. So we now know that although chemotherapy probably nowadays is less used in the clinic, but it is still used for some advanced HCC patients through transarterial chemoembolization administration or hepatic artery infusion. We know that patients that receive chemotherapy like 5-FU and cisplatin would actually enrich for a CD133 PROM1 or even other liver cancer stem cell marker expression. And here you can also see that in this mouse model, we're able to see an elevated PROM1 expression following 5-FU as well as cisplatin treatment, which is not shown here. Interestingly, if we deplete PROM1 cells, we were able to see that we can sensitize HCC tumors to 5-FU treatment and impede HCC tumor growth, as demonstrated by reduced liver weight and also the number of tumor nodules. Importantly, you can see that actually if we just use 5-FU alone, 5-FU did not reduce tumor growth because as you can compare the blue and the black. But importantly, 5-FU actually led to an elevated stem cell frequency, suggesting a higher chance of tumor recurrence if only chemotherapy is utilized. So because of this, we went on to try to understand, okay, so, again, what is the mechanism driving CD133 cancer stem cells? But unfortunately, although we proved the functional role of CD133 is actually not specific to HCC. It's also expressed in the regenerating liver. So to this end, we tried to compare the transcriptome profiles of sorted CD133 positive and negative cells harvested from regenerating liver that is induced by a DDC diet, or as well as HCC models, again, DNCCL4 or the NRAS-AKT that is delivered by hydrodynamic tail vein injection. So by this means, we were able to identify a number of deregulated genes, including one that is called SPINK1. Now, SPINK1 is actually a trypsin inhibitor, and in the normal physiological setting, it is actually almost exclusively expressed in the pancreas, where SPINK1 is secreted to protect the pancreas from autodigestion. And now, SPINK1 is, you can see, not expressed in the normal liver, nor many other major organs, nor immune cells, making it a very attractive therapeutic target. We were able to confirm by quantitative PCR our RNA sequencing observations that SPINK1 is overexpressed in the CD133 positive cells and also in the HCC tumor bulk in both HCC tumor models, but not expressed in the liver regeneration model, either induced by DDC diet or partial hepatectomy. We were also able to see SPINK1 to be elevated in the NASH-induced HCC mouse model, suggesting that its upregulation is observed across different etiology-driven HCC. Now, expression of SPINK1 and CD133 was found to closely correlate in both HCC models, and depletion of PROM1 would also lead to a drastic decrease of SPINK1, as confirmed by both quantitative PCR as well as RNA scope analysis. Considering that SPINK1 is actually identified as, from the CD133 cells, we thought that, it might be as closely associated with liver development. So we looked into various models. One of them is a fetal mouse livers at different developmental stages. And so we were able to trace it from a different embryonic stage, and we found SPINK1 to peak at between embryonic days 16 to 18, and then rapidly dropped during hepatocyte differentiation, which mirrored the expression of several liver progenitor markers, including keratin 19, keratin 6, SOX9, and alpha fetoprotein, which was published in our collaborators' PNAS paper in 2020. And then in a separate in vitro differentiation model in which human embryonic stem cells were differentiated into hepatocytes, using a defined culture medium and growth factors, SPINK1 was also found to be elevated in premature hepatocytes and then sharply dropped during hepatocyte differentiation. And then to further confirm this in a human setting, we also looked at some single-cell RNA sequencing data set that profiled human liver development. And SPINK1 expression was found elevated in hepatoblasts, fetal hepatocytes, cell clusters, but then markedly decreased in the adult hepatocyte cell clusters, with high SPINK1 expression correlating well with hepatic progenitor markers and not with the mature hepatocyte markers. And using cytotrace, which is basically a framework for predicting differentiation states, we also found high SPINK1, which is here, this cluster, to overlap with the less differentiated hepatic cells. And then further analysis in the TCGA dataset found SPINK1 expression to be progressively increased from well to poorly differentiated HCC tumors, with high SPINK1 expression clustering very well with the hepatic progenitor markers and not with the mature hepatocyte markers, and that it would also enrich for embryonic stem cell, undifferentiated cancer, as well as HCC stem cell signatures. High SPINK1 expression also was also found in patients that exhibited a high mRNA stemness index, and that using the tissue microarray, we were also able to see high SPINK1 to correlate with a worse survival, and also high SPINK1 in the NASH-related HCC human samples. So functionally, we were also able to show that SPINK1 plays a very important role in promoting self-renewal and chemoresistance. You can see here that knockdown of SPINK1 would reduce ability of the cells to self-renew, as demonstrated by tumor-initiating cell frequency. Knockdown of SPINK1 also diminished tumorigenicity in vivo, with a marked decrease in tumor incidence, and also tumor-initiating frequency, with an increased tumor latency and tumor-free survival than HCC cells expressing the non-target control. Knockdown of SPINK1 would also result in an increase in the ability of cells to resist chemotherapy in 5-fluorouracil, as indicated by increased rates of apoptosis. So we were able to also get similar results when we do an overexpression system, or if we added recombinant SPINK1, because SPINK1 is actually a secretory protein, or added a SPINK1 neutralizing antibody. We then also worked out the receptor for SPINK1, which turned out to be EGFR, and we did various co-IP, multiplex immunostaining, and also rescue experiments where we knocked out EGFR, but then added recombinant SPINK1, and showed that SPINK1 actually regulates HCC through an EGFR axis. And then we also worked out the transcription factor that would bind and transcriptionally drive SPINK1, which turned out to be ELF3. And again, using luciferase reporter assays and ChIP-qPCR assays, etc. Now, downstream, we were also able to show that SPINK1 would promote self-renewal, dedifferentiation, chemoresistance, and tumor initiation through a deregulated ERK, CDK4/6, and E2F2 regulatory axis. So how we started looking at this pathway was we found high SPINK1 to enrich for E2F targets. So we then worked upwards and saw that actually overexpression of SPINK1 would lead to elevated MEK, ERK, CDK4/6, cyclin D1, phospho-RB, and then also E2F2, but not E2F1 or the E2F3. And evidently, the first thing we would look at is cell cycle, because CDK4/6 and cyclin D1 is, would signify. But then, to our surprise, actually, cell cycle was not altered when we looked into that. And to support that, we also showed that the GSE, the pathway analysis of the HCC patients revealed no significant enrichment of the gene signatures that were associated with G1S transition or the cell proliferation in patients with high SPINK1. So when we did more reading, we also went and found out, actually, in addition to proliferation, the studies have also linked cell cycle to self-renewal and oncogenic dedifferentiation. For example, RB inactivation is found to promote the reprogramming of differentiated cells to a more pluripotent state, while cyclin D1 is found to be essential for pluripotency maintenance in human embryonic stem cells by preventing differentiation. So we found high SPINK1 expression to cluster well with E2F2 target genes that were related to stemness, dedifferentiation, and chemoresistance, including some of these include FGFR3, SPHK1, and MYBL2. And interestingly, if we look at EF3, SPINK1, and EU2F2 signature, high expression of these three would exhibit a worse prognosis than those HCC patients with low, drawing clinical relevance to further support this finding. So lastly, in this in this study, we try to look at SPINK1 as a possible therapeutic regimen as in a proof of concept model. So we treated HCC tumors with either chemotherapy alone or in combination with lentiviruses encoding SPINK1 knockdown, with the latter delivered intravenously into the mice. So lentivirus encoding SPINK1 knockdown and chemotherapy combination produce a maximum suppression effect as evidenced by tumor size, reduced tumor size. We have a suppressed liver weight and also prolonged survival time. Combination treatment also reduced tumor initiating cell frequency. And importantly, it also led to a significant decrease in stemness and also increase in differentiation gene expression. So in summary, we found this oncofetal protein SPINK1 to drive CD133 positive HCC towards a dedifferentiation progenitor lineage, and targeting SPINK1 may represent a novel therapeutic option for treatment of HCC, aiming at the CD133 cancer stem cell roots and overcoming chemo resistance. So this story is spearheaded by a now a final year project PhD student in my lab, Tony. So moving on, I also wanted to touch a bit on the liver cancer stem cell niche. So this again is from the article that we wrote, and we know now that liver cancer stem cell niche, including immune cells, of course, it's very important. Here, we know also that rapidly growing tumors often experience nutrient deprivation, and because of the poor vascularization in HCC tumors. So tumor cells often respond to these effects through trying to molecularly adapt to promote clonal selection of a more malignant cell phenotype. And we found that if we actually treat HCC tumors or HCC cells with glucose deprivation, it would enrich for a cancer stem cell population or a cancer stemness, including enhanced tumor initiating cell frequency. It would low glucose treatment would actually potentiate the cells to form tumors in vivo, and it would have elevated expression of CD133 and aldehyde dehydrogenase. Now, HCC tumors that were treated in low glucose would activate a cellular stress response PERK-mediated UPR signaling pathway to enhance ATF4 expression. And upon further analysis of the RNA sequencing data comparing the high and the low glucose, we actually identified a number of deregulated genes, of which FUT1 actually harbored 2 ATF4 binding sites on its promoter region. So FUT1, we know it actually encodes fucosyl transferase 1, which is basically responsible for the addition of fucose to glycans. So it's a post-translational modification. So analysis of TCGA data found HCC patients with to have elevated FUT1, and that high FUT1 would predict multinodular tumors. It would be correlated with advanced tumor stage and also worse survival. And further analysis in a different data set also showed that HCC patients with low FUT1 and aldehyde dehydrogenase to be more responsive to sorafenib treatment, suggesting that FUT1 together with the cancer stem cell marker aldehyde dehydrogenase may be a predictive marker for the response of sorafenib in the clinic. We also went on to try to understand the causative relationship between FUT1 overexpression and the functional role of FUT1 HCC. So we did a endogenous FUT1 knockdown in the immunocompetent HCC mouse model. In this case, this was an NRAS-AKT. And we showed that in this mouse model, FUT1 would be expressed from progressively from low to high, from early to advanced HCC. And then knockdown of endogenous FUT1 expression delivered through tail vein injection by lentivirus particles would attenuate liver to body weight ratio, as well as the number and size of the tumor nodules as compared to the controls. And then the ex vivo limiting dilution assay also showed that knockdown of FUT1 would decrease the tumor initiating cell frequency, which is indicative of stemness. So, so here in this story, we actually show that glucose deprivation would trigger ER stress response, and the ER stress response. So, elevated ATF4 would then bind to FUT1 promoter to induce its transcription. FUT1 as a post-translational modification would increase fucosylation modification on CD147, EGFR, EPHA2, and ICAM1, which would then translocate to the membrane and function as a membrane protein. And then in the latter part of that study, we also show that these fucosylated proteins would then converge into AKT mTOR signaling pathway to drive cancer stemness. So this was done and also published also later on in end of 2021 by Jane, who was a PhD student and now moved on to do her postdoc at Francis Crick Institute. Now, so for the last, I think maybe 10 minutes or so, I would also just switch gears a bit and talk about some of the things that my team has been trying to develop in the past few years. As we all know, HCC tumors is very difficult to treat, and pretty much the treatment for HCC patients is one size for all, and it's based on how advanced or how early the patient is diagnosed. And if we work with HCC tumors, it is very difficult to distinguish drivers from passengers, and models for more in-depth analysis of pathway interference in a defined mutation spectrum would obviously provide a more simplistic model for study. Now, now hydrodynamic tail vein injection is no news for everyone here, in particular in the liver cancer world, and so we also did the same and tried to establish driver-dependent mouse HCC tumors by hydrodynamic tail vein, and this is a few of the combinations that we did using CMYC along with a combination of either a tumor suppressor gene like AXIN1, P10, or TP53, or proto-oncogenes, including a mutated beta-catenin or an MCL1 overexpression. So in addition to profiling these HCC tumors by single-cell RNA sequencing and flow cytometry for immune cells and non-immune cells, we actually also created corresponding organoids with know-hows learned from Hans Cleaver's lab when we visited just for a short summer before COVID. So we were able to establish now a number of these organoids from these HCC mouse models, and that could be expanded for drug screening in particular. And we also actually did multi-omics analysis, including whole exome sequencing, RNA sequencing, ATAC-seq for chromatin accessibility, as well as proteomics and phosphoproteomics on both the tissue and the organoids for a deeper understanding of what kind of profiles will be altered in each independent driver-dependent mouse HCC tumors. So this is what the HCC tumors look like, and this is what the organoids look like. As we all know, it's a very good model to work with, because depending on what proto-oncogenes you inject, you're able to actually come up with HCC tumors within a few weeks, and then with the mice dying in 8 weeks' time, and with a very high penetrance success rate. So by whole exome sequencing, we were able to show that the organoids had a very clean mutation profile. So the organoids only retained, or basically the mouse tissue and the organoids only retained the driver mutations that were introduced into the mouse by hydrodynamic tail vein injection. So we see the CMYC overexpression for the AXIN1. We were only able to see the AXIN1 mutation, TP53, P10, et cetera, et cetera. And we were also able to show that overall whole exome sequencing shows a very much, it's not exactly the same, but it didn't show too much of a big alteration at the genomic level. Histological examination also found these HCC tumors to be derived primarily of hepatic origin. As we can see, there was abundant expression of HNF4 alpha as well as alpha fetoprotein. You can see that actually CK19, which is a marker of cholangiocytes, also was also picked up, but some of these were primarily in the non-tumor and not in the tumor models. We also did IHC analysis again to verify that whatever we injected was actually expressed, including MYC overexpression, depletion of the P10 and TP53, and overexpression of the beta-catenin and MCL1. So And so that was a tissue. And in the organoids, we were also able to show high expression of alpha-fetoprotein and HNF4 alpha, again suggesting that these models are representative of hepatic origin instead of the cholangiocytes. And the RNA sequencing data also showed that they were more showing a hepatic signatures compared to the cholangiocytes. Now, we went on then to try to compare the transcriptome profiles obtained from the sequencing of the mouse liver tissue and the corresponding organoids. And the first thing that we noticed was that the normal and the non-tumor tissue to group together and away from the HCC, distinct from the HCC. The second thing that we noted was that the tissue and the organoids to also group distinctly, which is not too surprising considering that, we do add quite a bit of growth factors into the organoids. But if we compare against the normal and the non-tumor, both the HCC tissue and organoids do show a decrease in the tumor suppressor gene, and as marked in the blue, and also an increase in the oncogene as marked in the red. And we will try to draw some clinical relevance to our mouse models, and we mapped each of these models to the various human gene signatures related to aggressive cancer phenotypes. And of these models, we were able to see that beta-catenin stood out to be the least correlated with proliferation, recurrence, stemness, and even a bit of survival. And this actually corresponded well to the molecular classification in human HCC, where beta-catenin is defined by G5 and G6 by the Boyle, or the S3 by the Hoshida classification, to be the most non-proliferative, displaying the least aggressive tumors, to be the most well differentiated, and also had a low frequency of vascular invasion. And so suggesting that, I think our mouse models do actually sort of correspond or recapitulate the human HCC subclass quite well. So if we dig deeper into the transcriptome data, we were also able to see distinct profiles between each driver-dependent HCC mouse model, as evidenced by the number of unique deregulated genes that were identified. But notably, we were also able to see common things that were picked up, in particular in the exon 1 beta-catenin and the PTEN models, and we're actually working now on trying to look at one of the targets that we also picked up through high throughput drug screening, so basically two ends did meet. So we did drug screening on the organoids, and the transcriptome data also corresponded well to show that it is deregulated, and hopefully we'll be able to show you more about this drug target and the pathway the next time I have an opportunity to share some of our work. So, for this part of the study, which is also still unpublished, HCC patients present high inter-tumor heterogeneity, yet conventional HCC mouse models fail to capture the high diversity, limiting mechanistic and translational studies. And here we have established and characterized at various omic levels distinct driver gene combinations induced mouse HCC models and their corresponding organoids. The models recapitulate human HCC transcriptional subclass, and we also now have some data, which unfortunately I'm not able to show, that this scalable mouse HCC organoid platform to be able to undergo drug response screening with some of the validation that we did in HCC patient-derived organoids as well. And so this is really a tour de force effort done by a number of teammates in my lab for many years. A lot of these had very good expertise with animal models, with bioinformatics, and also with flow cytometry. As I mentioned earlier on, we also did single-cell RNA sequencing and flow cytometry analysis and immune profiles, and which I'm sure a lot of people in the audience is also doing. So hopefully we'll have more to share on that later on. So hopefully I'm okay on time. Just like to end by thanking the people who actually did the work. It's really a pleasure to have such a great team that I work with, very motivated young students and staff in my team that share the same passion. I'm also very grateful to all my collaborators, my previous mentors, my mentors still now, including Spike Wan, and a number of collaborators now at Hong Kong U and overseas. And so with that, I'd like to end, and I'm happy to take questions if there are any. Thank you very much.
Thank you so much, Stephanie. This is a very fantastic talk with so much data that included, very interesting, sophisticated. So I think we have a time for questions. So I would like to invite, if you don't mind, you can just unshare the slides. And I will invite people who have a question. Perhaps you can turn on your camera and raise hand that will be called upon. So I would like to start with the first questions. You talk about the new features, the cancer hallmark as a plasticity, and then you also mentioned one of the genes that SPINK1 is somehow interested in driving the plasticity, that possibly driving the Stemness. So in a way that I think you show the data that I may missed it. Plasticity could reflect from either tumor cell plasticity as well as the tumor microenvironment. Do you have a sort of any data suggesting that how tumor cells from SPIN1 signaling that driving tumor microenvironment that somehow make those also plastic and then resistant to treatment, something like that?
That's a great question. How the SPINK1 tumor cells would modulate or not the immune, like just the tumor microenvironment, right? And change that to be more plastic as well, right? We have not, but we could possibly, let me think, we could possibly use the mouse model that we have with at the very end where we showed like sort of like a proof of concept therapeutic knockdown of the SPINK1 and try to look at the tumor microenvironment in that, right? And see whether anything has changed. So, but we don't have the data. We don't have that. But
you have you do have a single cell data, right? So have you explored the single cell data? Look at not only the change in tumor cell, but also change in the microenvironment.
You mean for the SPINK1 data? We don't have we don't have single cell data for SPINK1. We analyze, we don't have it for the single cell data for the SPINK1 mouse model, but we did analyze a few just publicly available data sets just to do correlation of SPINK1 with stemness and differentiation, but we don't have it with the mouse model. Yeah.
Okay. Let me see that we have one question from the chat box from Paul. Who's asked that, do you see any future clinical biomarker use of SPIN1?
Yes, thank you. We're actually working on that because we do actually, I didn't focus on that because there was a prior publication that was already published showing SPIN1 and use of it as a biomarker for HCC diagnosis and prognosis. So that's why in this work, we didn't dwell too much on that because it's already been published, right, by another group, not mine. But I think it will definitely be worthwhile to see if it would, in the context of chemotherapy, which is the focus of our study right now, for SPINK1 at least, to see if it would have any prognostic potential. And we're working with a clinical oncologist to look at SPINK1 in a more expanded sample size.
Okay, great. Tim has a question.
Yes, Stephanie, that was very, very interesting. My head is still kind of spinning around this. Now, I have a very simplistic question. So you show very nicely that depleting the cancer stem cells adds to the efficacy of chemotherapy.
Mm.
For instance, the data with the 5FU, etc.
Mm.
Now, what is the percentage of cancer stem cells in the tumor? And how do you explain this? I mean, why? I mean, what does it do? It's like, I mean, I know the number is going to be very low that you're going to tell me if but how does this work? Is this a direct effect, or, what do you propose? Why is 5FU working?
The very great question is something that I don't know how to answer. So the easy answer to your question regarding the percentage, I'll answer the easy part, which is how the percentage of cancer stem cells studies for CD133, it varies. So we did collect clinical samples from over 100 patients and looked at CD133 in particular, and it varies from 5% to 30-something%. So, and actually not necessarily the protein would match with the RNA sequence, the RNA data as well. So it's very tricky. But why would depletion of CD133 sensitize the tumors to chemotherapy, right? Possibly because we're depleting the tumor initiating potential of the cells, right? I mean, CD133 is just a surrogate marker, right? I mean, it's a marker, but it also has a functional role. But we know CD133 has this tumor initiating potential. So probably the 5FU would enrich, if we just use 5FU, it would enrich for CD133. But if we kill the cancer stem cells as well, then it would also deplete the tumor's ability to initiate initiate and also sensitize it to HCC and to 5FU.
So sorry to interrupt. So do you see this? So in other words, have you looked at after chemotherapy? Are there changes in terms of cancer stem cells?
So following chemotherapy treatment, there would be enrichment of a cancer stem cell subset, in particular for this CD133 expression, as shown in that SPINK1 data. So we were able to see a clear increase, actually, in the cancer stem cell frequency, as tumor initiating potential, as well as the CD133 population. And when we deplete it in the mouse model, this POM1, it would sometimes be more, it would be more sensitive to 5FU and cisplatin. Yeah.
Okay. Let me follow up another questions about the organoids. So you mentioned about organoid, but we our experience, many other also the establishment organoids from HCC clinical samples are quite challenging. So the success rate is extremely low. So the success was few of those. How do you see that those are representative of the whole patient population that you could actually model, use for study?
Same in our hands for human patient-derived organoids, very low success rate. We actually started off working, having very high hopes to try to create, patient-derived organoids biobank, but like for the gastric organoids or the colon. But our success rate in our hands was about 15%. And so we were able at the end, after like 2-3 years, to just manage to get 5-6 organoids from the patients that would grow, and these would usually be from a very advanced HCC tumors. So we were just able to grow a few to be used in our studies. But we try to apply what we know in growing these human patient-derived organoids in the mouse, and that turned to be quite successful somehow. And we did tweak the protocol a bit. But of course, with the help with Hans Clevers, and so growing those HCC mouse organoids is actually quite successful in terms of success rate. Yeah.
Okay, that's fantastic. So Carol has a question. Carol.
Hi, thanks. It was very interesting and fascinating, and it was also very intense, and I'm not a liver cancer person. So, but I was struck by a lot of the themes that are reminiscent for people who have been studying stem cells, or at least the concept of dedifferentiation throughout their career, like I have in another tumor model. But I was a little bit confused by the fact that, if I understood you correctly, your TDT tomato PROM1 positive cells actually had a higher proliferative capacity than the other cells, which is somehow, I think, counterintuitive to what's usually thought of as a stem cell. So can you put that in the context? I mean, is it perhaps a cell that's like more like a progenitor, and so you've captured this, and then the real stem cell is within one of your populations, a little less active, you know, and can.
Thank you, Carol. That's a very great question. And we did get a lot of questions from the reviewers regarding this, and we're still actually trying to figure this out. In fact, it is what it is. We were able to really see a increased proliferative potential. And so that's why at the end, we concluded that this may not really be the stem cells. Just that's why we called it tumor propagating cells at the end.
Right.
Because of this, you know, as you mentioned, right? Just repeating you, like the conflicting quiescent, actively proliferating concept, right? And in fact, in some of the stories, like the SPINK one, for example, we compared the CD133, right, with the HCC and the liver regeneration. We were also actually able to see, actually, people actually asked us, is your self-renewal a side readout of enhanced proliferative potential of those CD133 positive cells, right? Would it be just, you know, a side thing, right? And so we actually had to do a lot of things to try to inhibit the proliferation, but still be able to see a enhanced tumor initiating cell frequency. So going back to your question, I don't have the answer, but our data does show that, you know, CD133 does show proliferative potential. And I guess the best answer is that it is really not the stem cells, stem cells, but, you know, progenitor cells that's just more proliferative, that's all.
Yeah. So then let me ask you this too, then, because I'm interested in the experiments where you treat with the chemotherapy. So did you then see an increase in PROM1 positive cells after
Yes,
chemo?
we did. Yes, we did.
So, I mean, it seems sort of counterintuitive, because one always imagines that the
chemotherapy will kill the
proliferating cells. Right? Yeah. And perhaps it's the type of chemo one uses as well. So, I mean, I do think that's a different question. But thank you. It is very, very interesting. And all of us struggle with this idea, you know. I mean, we've turned to barcoding to try to get at this issue. You know, in neuroblastoma, we call them resistor cells or persister cells after chemo, and they do have a mesenchymal phenotype. So a lot of themes very reminiscent of, and the interesting thing in our model systems is that the surface expression, so we do a lot of CAR-T targeting, and the surface expression is quite different between the persisters and the bulk tumor, so that you may not get the stem cell if you use a particular, you know, therapeutic, a CAR-T approach and stuff like that. So they are confounders, and I think this is why liver cancer and many others are so difficult to treat, you know.
No, thank you, Carol. Yeah, I think barcoding, definitely. We're also trying to work out the CRISPR-Carlin system. We try to, you know, use the iPS-derived hepatocytes and transfect the Carlin to trace, you know, what is the you know, the events that turn on HCC, but it's been challenging for us. Anyway,
thank you very much. It was excellent.
Thank you, Carol.
Thank you. I think this is really speaking of how complex the cancer stem cell field, you know, topic is. Since over more than 20 years of research still continues, you know, debating about this side. So this is fantastic. Thank you so much, Stephanie, particularly in such a late time of the day for you, and I really appreciate you speak to this audience. Thank you. Let's give a round of applause to Stephanie.
Thank you.
Thank you, Stephanie.
Thank you.
Thank you, everyone.
All right. Bye.
Bye-bye.
So Stephanie, oh, she left.
She left, yeah.
Okay, hopefully she'll go on the next one.